So now uh, after knowing about the selection of plasmids or a vector, we'll see one by one which are the vectors which we can use for gene cloning. Okay, so one of that is plasmid. I have already shared one video with you. We'll see brief about this plasmids. Plasmids are defined as an autonomous element, or you can say extra chromosomal unit, which is not actually required for multiplication or reproduction for a bacterial cell or any other cell. They are usually easy to isolate, purified, and they can be reintroduced in a bacterium by transformation mechanism. Plasmids often bear antibiotic resistant gene that can be useful to recognize it, which are used to select their bacterial cells. When a recombinant plasmid containing this foreign DNA, it is also known as chimers. This is a brief diagram about this plasmid PBR322, which is very well popular and used as a plasmid vector. I will not go in much detail as we have already shared with video. Okay. But the important parts, as we have discussed in last lecture, that is ORIC, that is origin of replication. This is necessary in a vector so that it can be multiplied and we can get a number of copies. It has two resistant genes, which are ampicillin resistant and tetracycline resistant. Later on, this can be useful to identify whether it is recombinant or not. Means whatever we want to transfer, જે gene આપણે transfer કરવો તો એ transfer થયો છે કે નહીં એ check કરવામાં useful થાય છે. Plasmids are circular molecule of DNA that leads an independent existence in the bacterial cells. Plasmid almost allows carry one or more gene. Often these genes are responsible for a certain useful characters that can be appeared by a host cell. For example, the ability to survive in toxic chemicals, antibiotics like ampicillin, tetracycline, etc. Sometimes they carry some genes which gives an extra properties like some growth promoting hormones. They make and go for nitrogen fixation. They can carry, they can mobilize some molecules. They can solubilize phosphate. These are the characteristics which are carried out by plasmids. In a laboratory, antibiotic resistant is useful as a selectable marker to ensure that bacteria in a culture contain a particular plasmid or not. Means they are markers. Which markers, what we use for the markers for the identification of certain things. In the same way, in this plasmids, they are useful as a markers. Most plasmids possess that origin of replication, so they independently replicate in a cell. The smaller plasmid make use of host cell's own DNA. Sometimes it happens when there is a small size of plasmid, it can utilize the host cell DNA also. A replicative enzyme in order to make a number of copies. Whereas in some large plasmids, they have their own specific enzyme for replication, or they have their own replication process. A few types of plasmids are able to replicate by inserting themselves into bacterial chromosomes. This is another phenomena that plasmid itself become a part of that bacterial chromosome, and now they will start replicating. This integrated plasmid, they are known as integrated plasmid or episomes, and they are more stable and can be maintained uniformly. And in later on studies, you will see that episomes are more useful for such type of recombinant gene cloning or recombinant DNA technology. Just briefing again, plasmid PBR322 have both genes for ampicillin and tetracycline resistance. Several of these restriction sites occurs only once on a plasmid and are located within the antibiotic resistant gene. This arrangement is useful after the transformation process to detect it. This is our diagrammatic representation of plasmid PBr 322.